How pregnancy tests caused a global amphibian catastrophe. Chytrid fungus is linked to the population collapse of roughly 500 species of amphibians, with approximately 90 of those now considered extinct. Since its discovery, researchers tracked the fungus as it swept across the planet, infecting every continent, with the sole exception of Antarctica. But how did it spread all over the world? Well, a strong hypothesis says it was because of pregnancy tests. Before modern tests were developed in the 1970s, various unusual biological methods were used. Following the discovery of rising ACG hormones during pregnancy, it became common to inject a woman's urine into laboratory animals. In the 1930s, a method was developed called the Hogben Test, which relied on the African clawed frog. Urine injected into African clawed frog. If the woman was pregnant, frog ovulated. The Hogben test was a significant advancement because it was much faster. Results were obtained within 4 to 18 hours. Cost-effective. The frogs were easy to maintain, and unlike previous animal tests, the frogs were reusable. Accurate. It showed a very high degree of reliability. As a result, the large-scale exploitation and global export of African clawed frogs began, making the Hogben test the world standard for decades, until it was eventually replaced by non-animal immunoassay-based tests in the 1970s. So, what began as a medical revolution ended in an ecological disaster. Today we know the African clawed frog is resistant to Chytrid fungus. It is highly likely that exported frogs were asymptomatic carriers of the pathogen in their native habitat. When they were exported to medical labs and eventually released into the wild, they introduced the fungus to native populations that had no natural resistance. Subscribe to join us in the Wild Lab, and make sure you don't miss our next deep dive into the mysteries of the natural world.